is Nur Karmila Qistina binti Muhammad Rizal. I'm from AS1202A. My student ID is 2020-824-268. So without wasting any time, I'm going to present my study case entitled DNA Structure Flip. So for introduction, this study case is divided into 4 parts. The first one is setting the stage. Second one is discovering the structure of DNA. Third is building a model of DNA replication. And lastly, the last part is testing the model of DNA replication. So this is for BIO150. So let's answer the questions in part 1. So the first question is, what is the structure of DNA? So the structure of DNA is C, double helix. The second question, DNA is made of repeating units called C, nucleotides. So moving to the third question, the D in DNA stands for deoxyribose, as DNA stands for deoxyribonucleic acid. So for the fourth question, the bonds that hold the backbone of each strand of DNA together are called hydrogen bonds. So hydrogen bonds are between the nitrogen bases, and it holds 2 strands together. So answering the fifth question, the information contained in DNA is encoded by the so the answer is B. So it is encoded by the nitrogenous bases on the inside of the DNA molecule. Sixth question, for part 1, in which direction is DNA read? So DNA is read at 5' to 3' direction. Answering the seventh question, the letters in genetic code of DNA are A, C, G, T, and the A stands for B, adenine. So ACGT is an acronym for the 4 types of bases found in a DNA molecules: adenine A, cytosine C, guanine G, and thymine T. And last question for part 1 is, if one strand of DNA is ACGTA, the complementary strand would be D, TCGAT. So for information, this is because D is always paired with A and G is always paired with C. Moving to questions in part 2. So the first one is, why is understanding the structure of DNA and how it is replicated important? List some technologies and practical applications that could not have been developed without this understanding. So the answer is, understanding the structure of DNA helps us acknowledge how it is replicated, translated, and transcribed, and we can know how it is formed. These concepts go hand in hand, allowing us to further identify errors in transcription or how coding for protein works. And without this knowledge, technologies such as CRISPR-Cas9, stem cell research, and DNA testing such as 23andMe, and certain reproductive technologies could not have been created. So the second question is, how does the structure of DNA identified by Watson and Crick differ from the model previously proposed by Linus Pauling and the others? So their structure were completely different when Linus Pauling model had triple helix with the phosphate backbone on the inside and the nucleotides on the outside. Meanwhile, the Watson-Crick model had the phosphate backbone on the outside with the nucleotides in the middle. So the third question, what discovery by Erwin Chargaff helped Watson and Crick build their model of DNA structure, and how was this piece of information helpful to them? So Chargaff's realization that A equals to T and C equals to G combined with some crucially important X-ray crystallography work by English researchers Rosalind Franklin and Maurice Wilkins contributed to Watson and Crick's derivation of the 3-dimensional double helical model for the structure of DNA. It allowed them to know what percentage of each nucleotide makes up a particular section of DNA. So the fourth question, what is the main scientific weakness you notice in Watson and Crick's paper? So by today's scientific standards, this paper lacking a statistical analysis of their findings as well as general structure. So there is no p-value. Their background research is mostly criticizing of previous work by the scientists. So the fifth question, the conclusion of the paper published by Watson and Crick includes the now rather famous statement, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So describe what this statement means and why is it significant. So this statement refers to DNA's ability to be split into two and be used as a template to replicate itself. So this of great significance because it's the basis by which cells code for proteins. Without the ability to replicate, there wouldn't be the ability to translate and transcript, rendering the whole process smooth. Answering the sixth question, describe the specific contribution of each of the following researchers to the discovery of DNA structure. So the researchers are Francis Crick, Rosalind Franklin, James Watson, and Maurice Wilkins. So Francis Crick helped in the development of the helical diffraction theory, which was helpful in the understanding of structure of DNA. He also worked with James Watson in discoveries with the double helix structure of DNA. He also demonstrated that a group of 3 nucleotide bases on a single DNA strand codes for one specific amino acid. So Rosalind Franklin, using the X-ray diffraction, created images of DNA, which was used later by Watson and Crick to determine the structure of DNA. James Watson, he helped deduce the structure of DNA along with Maurice Wilkins, and it was his idea to use the X-ray crystallographic techniques to study DNA. For the seventh question, in their paper, how do Watson and Crick acknowledge Rosalind Franklin for the contribution for their work? So the answer is they mentioned that they were stimulated by the unpublished result of Dr. Wilkins and Dr. Franklin's lab. Part 3, building a model of DNA replication. So first, this slide shows the table of result based on the video given about the DNA replication. So answering the first question, describe what occurs in each of the following models of DNA replication: conservative, dispersive, and semi-conservative. So conservative, in this model, the DNA replication results in one molecule that consists of both original DNA strands identical to the original DNA molecule, and another molecule that consists of two new strands with exactly the same sequences as the original molecule. In the dispersive model, DNA replication results in two DNA molecules that are mixtures or hybrids of parental and daughter DNA. In this model, each individual strand is a patchwork of original and the new DNA. And semi-conservative, in this model, the two strands of DNA unwind from each other and each acts as a template for the synthesis of a new complementary strand. This results in two DNA molecules with one original strand and one new strand. Next question. Describe how each model of DNA replication differs in terms of the observations you made using gumdrops and marshmallows. How can you tell each model apart based on the relative amount of gumdrops and marshmallows in each DNA molecule. So the answer is in the conservative model, there would be either 100% gumdrop or 100% marshmallow, meaning there would be dense DNA at the top at the bottom of the tube and less dense DNA at the top of the tube and nothing in the middle. In this disperser model, there would be 50% gumdrop and 50% marshmallow in each strand of DNA. There would only be a strip of DNA in the middle of the tube. In semi-conservative model, there will be DNA with 100% marshmallow or DNA with 50% gumdrop and 50% marshmallow. And there should be a strip of DNA at the top of the tube and a strip of DNA in the middle of the tube. Moving to the multiple choice questions in part 3. So, which of the following statements about the use of gumdrops and marshmallows in this activity is true? So, the answer is A, that both are used to represent nucleotides. So, second question, in this activity, DNA molecules containing only original template DNA represented using A, gumdrops and toothpicks only. So the third question, after one round of replication, what has been produced by the conservative model of replication? So the answer is A, one molecule that contains 0% gumdrops and 100% marshmallows, and one molecule that contains 100% gumdrops and 0% marshmallows. So the fourth question, after 2 rounds of replication, which of the following is true? Of the 4 DNA molecules produced by the semi-conservative model. So the answer is C, 2 contains 50% gumdrops and 50% marshmallows, and 2 contains 0% gumdrops and 100% marshmallows. And the last question, which models of DNA replication never produces a DNA molecule that contains both gumdrops and marshmallows? The answer is A, the conservative model only. So moving to the last part of the study case, the question is, before analyzing the result of this experiment, Michelson and Stahl needed to know where DNA containing 100% 14N or 15N would be in the density gradient. So predict what Michelson and Stahl did to figure this out. So the answer is they grew the DNA in a 14N and a 15N both separately. So moving to the next question, how can you interpret the data collected after one round of division? So in other words, what do the number and location of the bands present signify? So the answer is there was half 15N DNA and half 14N DNA. Next question, which models of DNA replication are consistent with the data collected after first round of replication? Which models can be ruled out and why? So the first answer is semi-conservative and disperser models are consistent with the data collected after the first round of replication. In conservative, the DNA settled in the middle of the tube, which rules out the conservative model, since in the conservative model, the DNA should be at the bottom or top of the tube, not the middle. So moving to the next questions. For any models of DNA replication that were ruled out after the first round of replication, what would the data have looked like if the DNA had in fact been replicated according to that model? So the DNA should have settled on the top of the tube or the bottom. Next question. What would the data have looked like if the DNA had in fact been replicated according to the model just ruled out? So the answer is a strip of DNA in the middle only. So moving to the last question. According to the model of DNA replication identified as correct, what would the data look like after a third round of replication, and how about after the fourth round? So the answer is there would still be a small strip of 14 or 15N mixed DNA, and the rest would all be at the top of the tube, since it would be the less dense 14N DNA. Last, why are bacteria like Escherichia coli good for studying DNA replication as opposed to using other types of cell? So it is because of its simplicity and its rapid growth.